Also, will this mic be on?
It will be.
Okay.
So
that is
if it's tilted sort of towards the height of your head, that's good.
Okay.
We're about the same height, so. After this conference, where are you headed?
I'll be here tomorrow. Oh, entirely. We'll start work at Lincoln Labs.
Yeah.
Next week, actually. Yes. So I'll be there for a year, and then the current plan is to go to NYU for PhD.
Okay.
Yes. Working with Eros and each other.
Yeah, amazing. Yeah, a good friend of mine, Alex Bergino, is in the room with that group.
Not a familiar name.
No, but you'll see it on
Yeah.
the website. Be a fun person to talk about. Yeah. That's great. That's very exciting.
I will also be here tomorrow.
Yeah. Good.
Where are you off to after this?
Family back home. I've been traveling a lot the past few weeks, but I'll be headed back to California to do my laundry.
A good goal.
Yeah, and starting for the year. Okay. All right, everybody, if you could find your seats, we're going to start the first talk session of CCN. Great. Okay. My name is Judy Fan. I am an assistant professor of psychology at Stanford University, and I will be chairing the talk session unfolding over the next hour. There'll be 5 talks. And they'll be pretty tight. So we've allocated about 10 minutes for each talk, including questions. So we'll really only have time for maybe 1 question after each talk. So if you have one in mind, please come to the microphone at the front so that you will be sure to ask your question, okay? Our first speaker is Nishka Pant from Brown University. Take it away.
Great. Thank you, Judy. I'll be presenting a work on HMAX, as we're talking about self-supervised learning of human-like scale invariant representations. So let's begin by taking a brief look into the past. From the late 80s through the early 2000s, models of the visual system were heavily constrained by anatomical and neurophysiological data. These models were trained with Hebbian-like unsupervised learning mechanisms, and they often had explicit hierarchical pooling mechanisms for invariance to object transformations, such as position or scale. The HMAX is a representative model of that class of feed-forward hierarchical models. Moving into the present, in the era of deep neural networks, we have a myriad of models that have achieved superhuman performance levels on a multitude of tasks. These models tend to be trained with supervised learning mechanisms using gradient descent, and they are developed without biological constraints in mind. In this vein, we develop these models without explicit hierarchical pooling mechanisms to invariance, to object transformations, except position and some CNNs. Instead, we learn invariance through data augmentation techniques. These modern deep nets and CNNs are really just elaborate versions of these earlier models. Additionally, these models that are developed without biology in mind tend to outperform these classical models on metrics such as brain score. And so you can see on this plot here, we have, as we increase in ImageNet accuracy, we are also seeing an increase in brain score. So our models from the past have been entirely superseded by these task-optimized deep models. But can they strike back? Can we still do well with these models of biology? And so let's try. We'll start out with the basics with an MNIST data set. And so to create this data set, we started with a central reference scale. We then upscaled and downscaled this data set 8 times to create a 17-scale data set used to verify our properties of scale invariance in multiple places. So I'll refer back to this data set quite a lot. Okay, so let's take a look at how our deep neural networks are doing on scale invariance tasks. Let's start by looking at a ResNet. This ResNet displayed here, as the legend shows, was trained only on the reference scale. This graph displays its performance at test time across the entire scale range, from a scale factor of 1/4 to a scale factor of 4. As we can see, this model performs well on the reference scale, which it saw during training, but performance drops off steeply on scales that were unseen during training. If we train the model on a partial segment of the scale distribution, from a scale factor of 1/2 to a scale factor of 2, again, we can see at test time we perform well on that scale range, but as soon as we reach scales that were unseen during training, we again witness a drop-off in performance. As one might expect, as if we see the entire scale range during training, at test time we perform well over the entire scale range. We see a very similar pattern in VGGs. This is the same experiment done with a separate model. And so what we get from here is that invariance is really learned in these models from just seeing what it has already seen before. So we are invariant on what we've already seen. However, if we are outside that distribution, we do not witness the scale invariance. Unlike what we see with these DNNs, there is evidence that humans can learn to recognize objects at scales never seen before during training. So this psychophysics experiment was developed by Han et al. In 2020, where participants were asked to see a target character followed by a test character. These target character and test characters were multiple different sizes, either 30 minutes of visual angle, 2 degrees, or 5 degrees of visual angle. The test character could either be the same as the target character, or it could be a visually similar distractor character. And participants were asked if the characters were same or not the same. So this was a discrimination task. Over here, we can see the results of non-Korean participants. This is important that they were non-Korean because they do not have prior familiarity with this data set. Across the bottom here, we can see the axes are defined by the size of the target, followed by the size of the test character. We see that these humans are almost completely scale invariant on this task, even though they have not seen the stimuli beforehand. In ML jargon, we would call this out-of-distribution generalization. If we look back at these DNNs that we said were scale invariant over the whole range, so the ones that we trained on the entire scale range during training, they do not generalize well to this task, so they cannot discriminate between the stimuli in the same way that humans can. So let's go back to our classical models. The HMAX had a mechanism for pooling not just over position like in CNNs, but also scales and potentially other affine transformations such as rotation. This mechanism we called scale bands. Each band is composed of filters that activate for features of a particular size. Smaller features might not necessarily activate larger scale bands, and larger features may not necessarily activate smaller scale bands, but if you pool over these bands, you should have the same amount of activation going into later layers of the model, regardless of the original size of the feature. For those of you that are familiar with the idea of max pooling over position for translation invariance, this is something very similar. So we want to improve this model. We don't just want to use the old version of the model. We rewrote this in PyTorch, and we incorporated trainable filters to increase the capacity of the model. The brain and the original HMAX resized the features and the filters and feature templates explicitly, but instead of doing this, we found that it is mathematically equivalent to have a fixed filter size and vary the size of the image instead. And so that is what we chose to do for efficiency. Now with this model, the naive thing to do would be to train this with backpropagation and cross-entropy loss, so that's what we started with. To explain how the model, we'd expect the model to work, if we saw an input of this size, which corresponds to the second scale band on this diagram here, we would expect this to be the maximally activated path through the model. Similarly, if we have a larger input, we would expect a larger scale band to be maximally activated. So when we took a look at which scale bands were being maximally activated in the model, across the bottom here we see the scale of the input, and across the side here we see the scale bands. What we would expect is we would expect the maximal activation to show up very strongly diagonal on this chart. However, we do not see this. We see some amount of interference, which indicates that for a particular input, the corresponding scale band is not being maximally activated. To combat this, we introduced another term to our loss. So what we did is we ran through the model at the same time our input image and a slightly rescaled version of that image. When we got to the penultimate layer of the model, we took the mean absolute error of the representations from that layer. And our idea here is to enforce that those representations are the same, so we're trying to minimize that mean absolute error. The CE term up there is also the normal cross-entropy loss. And so with this contrastive loss mechanism, we see that we are doing a much better job, the model, of picking the correct scale band for the corresponding input. So let's look at how we are doing on the scale invariance task that we originally tested on. These are the results from the deep networks that we were looking at. Our HMAX trained naively with backpropagation does pretty well, but we do see a small drop-off on the smaller scale sides. And our contrastive HMAX seems to perform well across the entire scale range and matches what we'd see from deep neural networks that are trained across the entire scale range. On our Korean task, we see that our HMAX with backpropagation doesn't do too well on this. However, when we introduce this contrastive loss, we are seeing a match in human performance. And what we can infer from here is that our contrastive loss HMAX is actually learning scale invariant feature representations, not just rote memorization of the data set that it saw before. So some ongoing work that we're working on here, we're looking at neurophysiology data presented by Pasupati et al. In 2016, where they have shown that for a neuron attuned to a specific orientation, the activation of that neuron will be the same regardless of the size of that feature. And we're also taking a look more deeply into some brain score data showing that the CHMAX potentially does better than other models on brain score when we test correctly for scale invariance. We're also doing some preliminary work on ImageNet. On the bottom here, you can see that we are finding that our contrastive loss HMAX, a slightly modified version of the model, is using the same features at different scales to recognize what the object is, as opposed to some of our models on the top that look like they are using different features at different scales to recognize what the model is. So, in conclusion, we've provided a version of the HMAX trained with backpropagation and one that's trained with contrastive loss. The CHMAX exhibits scale invariance, and it generalizes well to data that we've never seen before during training. It is also able to select for scale and filter size correctly. And we have also provably shown that learned scale invariant CNNs do not generalize to data that has not been seen before during training. I know that was a lot of information very, very fast, but if you have more questions, you can also please come find us at our poster tomorrow, which is B64. Thank you.
We do have time for one very quick question, if there is one. In the meantime, we'll also set up for our second talk.
Hi, great job with the talk. I'm just wondering, have you ever looked at how modern vision transformer models compare to these ResNet models, traditional DNN that you have compared to? Because to me, transformer vision models could be inherently more robust to scaling issues.
Sorry,
the attention mechanism.
Could you rephrase the question?
Have you looked at transformer vision models and their performance in terms of how robust they are to the scaling issue that you're trying to solve?
Yeah, so we very briefly looked at transformer models, not super in depth. We have found similar drop-offs in performance, or like failure to generalize outside of the scale range in some preliminary experience, but that's definitely something that we're planning on looking into more, especially given the prevalence of transformers these days. Thank you.
All right, thanks so much, Nishka. Our second speaker in today's session is Jean-Nicolas Jérémy from Aix-Marseille Université. Take it away.
Thanks. Okay, thanks. So I'm Jean-Nicolas Jérémy, a PhD candidate under the supervision of Dr. Emmanuel Dossé and Laurent Périnet, working at the Institut de Neurosciences de la Timone. And today I'm going to show you a part of my thesis work on how retinotopic and CNNs, convolutional neural networks, implement efficient visual search. So this work relies on a neuromorphic approach, where we are trying to take inspiration about a biological function, retinotopy, to improve tools or methods, CNNs, for tasks, so visual search, which is here, the localization and the categorization of an object of interest. So first, what's retinotopy? Retinotopy is the way the visual field is encoded in the human brain. So here we have a nice figure from the Dugolci and al. Work, which display the fMRI activation in the human brain when displaying different stimulus, either in eccentricities or in angularities. And we can see that both of these characteristics are encoded orthogonally in the human cortex. So there is also an overexpression around the point of fixation. You can see these red rings is overexpressed in the cortical areas where it takes more than half in the space. So this creates a new referential framework where the eccentricities and the angularities are encoded orthogonally with this overexpression around the point of fixation. And this is known for a long time, so we know how to modelize that, and we can use the log polar transformation to model this. So to illustrate this transformation, here is a grid picture in the Cartesian space with the x and y axis. And if we apply this function, the log polar transformation, and we reattribute the coordinate of each pixels following this new referential, we have one axis which is angles, thetas, and the other one which is rho, the eccentricities. Also, we add a log function to mimic the overexpression, the overrepresentation around the point of fixation. We can see that the gray square come from 1/9 of the surface to merely half of the surface, which mimic the fovea in the human brain, a high-resolution place in opposition with the periphery where the information is compressed. So another important feature is that rotation is a variation in angles and zoom is a variation in eccentricities. So these transformations in the Cartesian space are assimilated to translation in the log polar space. And this is interesting because we are working with CNNs which are known to be robust to translation. So in order to confirm this hypothesis, we're trying to train ResNet 101 networks either on Cartesian or in a retinotopic frame. And we test those networks on the ImageNet dataset with 50,000 images, the validation dataset, and 1,000 labels. In blue is the Cartesian networks, and in red is the retinotopic one. On the left, you can see the average accuracy across different angles and for different zoom ratio. And without any rotation, you can see that the Cartesian networks have an accuracy around 83%, but this accuracy drops when we switch to another range of angles. And the same effect is observed with the zoom ratio, while the retinotopic networks has a slightly lower accuracy, around 78%, but it remains at this accuracy around the different range of angles or with the zoom ratio. So this research can help us to define the effects of this assimilation of rotation and zoom in the log polar retinotopic frame, and that brings robustness to the networks. So now we saw this transformation, rotation and zoom in Cartesian. What happened when you are doing a translation in the Cartesian space? So translation for in biology is moving the fovea, and when you're moving the fovea, you're doing a saccade. For the Cartesian space, you can see that if you focus on the object of interest, the leopard, you can see that between these 2 points of fixation, the surface occupied by the leopard is twice greater in the second point of fixation. But in the retinotopic space, it's because of the compression of the information at the periphery, these same leopards come from around 5% to around 50%, so 10 times greater. So this transformation is highly sensitive to translation. And we infer that this characteristic is an important feature to categorize, localize efficiently an object of interest in the space. So we built a likelihood map protocol in order to test this hypothesis, and we display a grid of point of fixation in images and feed the networks with all these images and ask the networks if they are not an object of interest. You can see some examples for the Cartesian and retinotopic networks. And you can see that it's the focus is the retinotopic networks maps are more focused around the object of interest. Also, we measured that effect by computing the activation ratio between position inside the region of interest versus outside those regions over the whole ImageNet datasets. And we can see that the retinotopic has a 41% increase inside the region of interest compared with the Cartesian, which is 23%. Also, we displayed the mean likelihood of the networks as a function of the distance to the max position. And we can see that the mean likelihood is higher for the retinotopic networks, and then the slope is sharper to the minimum. All of this together show that the retinotopic version has a better contrast in the heat maps, likelihood maps. But okay, the contrast is okay, but we need to locate efficiently the object of interest. So let's get back to the first picture where we display the heat maps for the Cartesian and retinotopic networks, sharper for the retinotopic, as we saw. And now, if we highlight the point of fixation with the highest likelihood, we can see that because the map is finer, we have more chance to be located on the region of interest. And we also measure that in the whole ImageNet dataset by computing the pointing game, which is an accuracy of the localization based on the most cyan point. And once again, the retinotopic version is better than the Cartesian one, with a 76% accuracy against 66%. So all these results show that retinotopic version seems to be better to locate efficiently an object of interest. And this work took place in a much bigger project where we, in the same neuromorphic approach, we try to modelize the dual pathway in the visual primates. And we have what and where pathway. This works will be the where pathway, where from a large display, we use the retinotopic local input to produce likelihood maps that highlight the region of interest, and we can then feed this position to a saccade system and then perform the localization, the categorization. Because CNNs are known to perform great in categorization, we focused on a good localization, a good and robust localization. So thanks for your attention. I will be presenting my poster tomorrow, and you can flash this QR code if you want the preprint of the paper, and open for discussions and everything. Thank you.
We have time for a question?
How does this work when you have two eyes?
One more time?
How does this work when you have two eyes input onto the cortex?
Oh, I'm not sure about that. I believe you can. The two eyes are bringing more the precise representation of the visual field. But still, afterward, in the visual areas, we have a single representation of the whole space. And you can just use both inputs in order to make better saccades in the object of interest.
All right, thank you, Jean-Nicolas. Our third speaker of the session is Kathy Garcia from Johns Hopkins University.
Hello, thank you. As Professor Fan said, my name is Kathy Garcia. I'm a PhD candidate working with Leila Ishik, and I'm really excited to be sharing with you all some of my work. Modeling dynamic social vision highlights gaps between deep learning and humans. In the last decade, there has been amazing progress in modeling the computations in the visual cortex, thanks to the advent of deep neural networks, which not only provide state-of-the-art performance on many vision tasks, but also match brain responses throughout the visual cortex. Now, we have a wealth of models, some of which are shown here on the x-axis, that are very close to the noise ceiling in terms of explaining variance in brain responses. However, these have been evaluated almost exclusively on static scenes with very little social content. But the real world, as we know, is highly dynamic and focuses a great deal on other people, like their actions and their interactions. And recent work has suggested that in addition to the ventral and the dorsal stream, There's a third visual stream along the lateral surface of the brain, specialized for processing social motion, often referred to as the lateral stream. Now, recent work from our lab, led by Emily McMahon, curated a dynamic data set of 3-second naturalistic social videos to map the responses in this pathway. In total, we had 250 naturalistic two-person videos that were shown to participants without sound in the fMRI scanner. They also collected behavioral ratings on these same videos. Then, they compared the behavioral ratings to the fMRI data. Much like the ventral stream, they found the lateral regions of interest with low-level visual and motion features in early regions, then mid-level information about the configuration of bodies, and finally, they found in the most anterior regions represent information about social interactions in the videos. But unlike the ventral stream, these regions have not been compared to DNN responses. Now, in our work, we wanted to look at how different types of models perform on predicting the human responses to the videos. So we asked, first, can DNNs model human responses to dynamic stimuli? To answer this, we use language, image, and video models to predict the behavioral responses. Next, to try and understand the computations in the lateral visual stream, we benchmarked DNNs against the human fMRI data from the lateral ROIs to evaluate the DNN performance. Finally, we asked across all the models tested, are there any computational factors that can improve lateral stream predictivity? Now, we evaluated the performance of the DNN models on the neural data and behavioral ratings collected from the original videos. To test the language models, we also collected sentence captions for each of the videos. The videos, the representative frames, and the captions are then input into a DNN model, and the features are extracted from each layer of the DNN. The extracted features of the DNN are then used as predictors to fit the observed responses. Then we can compare the predictions to the actual neural responses. But we didn't just do this for a single model. We tested over 350 image, video, and language models, including state-of-the-art models. Now, first, we look at the DNN performance across the image, video, and language model classes in predicting the human behavior ratings in the videos. So here on the x-axis, I'm showing each of the different features, and on the y-axis, we have the encoding score for the best performing layer in each model. Here, we click the best score for each of our over 350 models, where each model is a dot, and the solid bars represent the mean score for each model class. And here, first, we're actually looking at spatial expanse. Sorry. At spatial expanse, which indicates whether the video takes place up close or far away. This is actually a standard scene feature, and all model classes perform here equally well at the level of noise ceiling, which is shown here in the gray bar. Now, for the mid-level features, like whether an action is directed at an object or whether the people are nearby or facing, all model classes perform similarly, but now are no longer at that level of noise ceiling. For most of the high-level social features, for example, if people are communicating or valence and arousal, we see that the best performing model class is the language models, despite the overrepresentation of image models in our set, suggesting that language models capture language embeddings of these sentences that current visual vision models possibly cannot. So next, we ask, how well do these models predict brain responses in the lateral stream? This time, on the x-axis, I'm showing each ROI on the lateral stream arranged from posterior to anterior. Although previously we saw overall language models perform best in predicting behavioral responses, here in the brain, they are far from the noise ceiling. While the image models do better in the EVC area, they are similarly not great in mid and high-level regions. In contrast, the video models here show a meaningful difference in performance. Now, actually, if we take a closer look and compare our image and video models, we can see here that dynamics provides a clear advantage in performance, especially in the mid-level regions like MT and EBA, with dynamic video models performing much better than static image models. While this may seem unsurprising, as prior research indicates, the lateral stream responds mainly to dynamic stimuli. However, most of the video models in our set are actually a few years old and are considered mediocre from a computer vision point of view, and especially compared to some of the powerful and state-of-the-art image models tested here. Additionally, as a baseline, we can also look at PPA, a ventral region known to process static scene features. And here we see that in contrast, performance here is very high, almost at the noise ceiling for some of the models. We can also see that the advantage to dynamics is specific to the lateral stream. Finally, I want to highlight that none of our model classes, even the video models, are doing a good enough job in predicting responses in the STS, the anterior region specialized for social perception. So, can DNNs model responses to dynamic visual stimuli? The answer is yes. DNNs are above chance, but they don't perform as well as they do in the ventral stream. And next, in terms of computational factors, the answer is yes, and it is dynamics. We saw clear evidence that video models outperforming image models in the lateral stream, suggesting that the insights from video models could be critical in improving DNN performance on dynamic social vision. Now, going across all of our results, the main takeaway here is that no single model could do a good enough job of predicting lateral brain responses, especially in the STS, and no model could predict both the neural and behavioral responses equally well. And with this, we reveal a huge gap in current AI models' ability to match human visual behavior to dynamic social scenes. In all, I think this truly highlights the need for further study and model development in dynamic social vision. I'd like to thank my first co-author, Emily McMahon, and my lab mates at the JHU Computational Cognitive Neural Lab, and a special thank you to the following scientists who made this work possible. Thank you all for joining me on this talk. If you'd like to check out our completed work, you can scan this QR code under the same title. Also, not up here, but our poster session, we have one tomorrow. Thank you all again.
Any questions? Yes, you take her.
Hi, very nice talk. My question is, did you differentiate between the dynamic component and the social component? Did you have any control dynamic scenes that you contrasted?
I'm so sorry, I'm having trouble hearing your question.
Sure, sure. So my question is, did you differentiate between the social component and the dynamic component? Did you have any control dynamic scenes that didn't have any social component in it?
Yeah, so all our videos are specifically social videos, but are you asking specifically like other scene features?
I guess my question is the model failure due to it not being able to model the social component, or is it not able to model the dynamic component of the scene?
Yeah, I'm not sure I understand your question, but I can follow up with you. Yeah.
Thank you so much, Kathy. Our next speaker is Professor Guo Jiahui from the University of Texas at Dallas.
Thank you so much for the introduction. Today, I will talk about shared connectome and organization in the human cortex, irrespective of sensory experience. So we know our human halo-visual cortex has many regions which are selectively responsive to different categories of information. For example, these two brains, they show the regions which are selectively responsive to faces. And these two brains show the regions which are place selective. Whether those regions need earlier sensory input to normally develop has long debates in our field. For example, people who used the monkey deprivation study suggested face-selective regions, they need those information to normally develop. However, people who did human research found category-selective activations using tactile or auditory stimuli, suggesting those regions may not need those earlier sensory input to emerge. In order to localize those regions, previous study, researchers used scans called localizer scans to complete this task. For example, the left figure shows an example of the classic category selective localizer used in the previous studies. By doing contrast between one category versus some of the other categories, we can localize those regions which are selectively responsive to a specific category. For example, the right figure shows the regions, shows the contrast between faces and other categories. However, those localizers are often vision-based and which are not suitable for people who cannot see. In order to overcome this problem, our lab, we developed a method which is based on hyperalignment and naturalistic stimuli. In detail, we can calculate the transformation matrices from all brains in one group into each brain in another group. And then we can apply those transformation matrices to and project the localizer data from all brains in group 1 into each brain in the group 2, and then calculate the mean topography. For a comparison, and we can also calculate the mean surface aligned topography as a baseline. Here, we can see the individualized topography can be predicted with high fidelity using movies. The second column shows the results we got by using connectivity hyperalignment, and we can see the predicted topography looked very similar to the maps we got using a localizer. And it's individualized and preserved those specific features for an individual. However, the maps we got using surface alignment is blurry and didn't preserve any of the individualized information here. This method can also generalize across different situations, such as movie contents, languages, scanners, etc. Based on this method, we can use naturalistic stimuli, which has many different modalities in order to match the specific needs for the sensory-deprived special groups. In this study, we used a dataset called 1-1 Dalmatians because everybody in this dataset watched this movie. This data was collected by our collaborators in Italy. And this dataset has 3 different normal participant groups. They either had both audio and visual inputs of the movie, or only had the audio input of the movie, or just the video input. And we also had 2 separate sensory-deprived participants groups, one congenitally blind participants and one group of congenitally deaf participants. They can either hear or see. We also used another 2 independent datasets, Budapest and the Monkey Kingdom datasets, in order to predict and validate our findings. These 2 datasets were collected at Dartmouth College. The beauty of our method is we can predict the regions using a dataset which was collected in a totally different country with a different language. These 2 datasets has both movies and a dynamic localizer included. Our analysis steps has 2 big parts. In the first part, we used everybody in the Budapest dataset to predict the topography for the participants in the 1-1 Dalmatian dataset. And the second step, because the 1-1 Dalmatian dataset doesn't have the localizer, we introduced the Monkey Kingdom dataset in order to validate our findings. So let's focus on the results we got from the first prediction step. We will use the face-selective topography as our example here. We can see for the people who had both audio and visual input of the stimuli, their predicted topography aligned well with our prior knowledge. The regions are along in the ventral temporal cortex, along STS, and in the frontal lobe. For the people who only had audio or visual input of the stimuli, their predicted topography looked very similar to the people who had both inputs. Most importantly, for the people who had no prior sensory input, blind and congenitally blind and deaf participants, their predicted topography, especially for the blind participants, looked very similar to the people, our normal participants, which suggests those regions may not need earlier sensory input to emerge. You may ask, probably your predicted topographies are blurry and doesn't have any specific features. This is not true. We randomly showed 2 participants in every group, and we can see their individualized topographies preserved those distinctness for the individual features. The shared functional topographies are not only true for the face category, and for the body, places, and object selective categories, the congenitally blind participants had very similar predictions as in our normal participants too. Now let's focus on the second step, validation. In detail, we got predictions of the individualized topography for everybody in the monkey kingdom dataset, and then we correlated the maps we predicted to the maps we just got using their real localizer. This step measures the performance of our prediction, and this also validates whether we can take the maps we got in the 1-1 Dalmatian dataset as if they were derived using a real localizer. The second step of the validation is a control condition. We shuffled the individualized topography across participants in the 1-1 Dalmatian dataset and did a similar prediction and correlation. Now let's see the results. First, we can see like the y-axis is the correlation is the performance, and the red line is the noise ceiling, and the gray line is the baseline based on surface alignment, and every bar is a group. And we can see our prediction is well above the baseline and not far away from the noise ceiling. And if we destroy the individualized correspondence, shuffle the maps across individuals, our prediction roughly dropped to the baseline level, which means our predictions is accurate and also participant specific. This is also not only true for the faces, but also for the other 3 categories. Now let's do a summary here. First, we find individualized topographies can be predicted in the congenitally blind and deaf participants, although they had never had any earlier sensory input, similarly as in the normal participants. Our study suggested development of the functional organization of the human high-level cortex can occur independently of prior sensory experience. I will give all of my thanks to our collaborators and funding agencies, and we will have a poster session on Thursday at 1:30 p.m. And the last note is I just started my lab at UT Dallas, and I'm recruiting trainees at all levels. And if you are interested, please contact me. Thank you.
We have time for a question. Okay, well, there will be plenty of time for questions. Please follow up with Guo Jiahui in any of the upcoming breaks. Now we will move on to our last talk of this morning's session by Sarah Harvey of the Flatiron Institute.
Okay, thank you for the opportunity to speak about our work on the duality of burrs and shape distances. And so this project really revolves around the question of what does it mean to compare neural representations. So suppose we're doing an experiment with some mice that are exposed to various stimuli. And we'd like to know if there's some learned representation that is general across mice or if there's large individual variability. And our question is then, how should we quantify mathematically how similar the neural representations are? And so we could imagine recording neural responses from each mouse and stacking these responses into a matrix that's stimuli by neurons. And even visualizing the neural responses as a set of M-labeled points in a high-dimensional neural firing rate space, or even as potentially lying on a high-dimensional neural manifold. And so we could ask, how similar are these representations in the high-dimensional space? And so this problem occurs across neuroscience and machine learning contexts, whether it's measuring the degree of similarity between multiple individuals, between multiple artificial neural network representations, perhaps after some change in training data, or initialization, or architecture, or even evaluating the alignment between artificial models to biological systems. However, there's an obvious problem with this framework because the neurons in each network, especially in the biological case, are basically sampled randomly and are arbitrarily indexed. So for instance, just subtracting these matrices is definitely not meaningful. So figuring out how we should measure representational similarity in a meaningful way is a non-trivial mathematical problem. This has sparked a proliferation of methods, which we noticed often fall into two categories. The first is similarity measures that transform or align neural dimensions by fitting some alignment transformation or nuisance transformation, and then measuring the distance between aligned data matrices X and Y. And this category contains, for example, techniques like linear regression, canonical correlations analysis, Procrustes shape distances. They generally take the form of distances on neural representation matrices X and Y. The second category contains similarity matrices that compare stimulus by stimulus matrices of summary statistics that capture pairwise similarity between stimuli. And this includes techniques like representational similarity analysis, centered kernel alignment, and the normalized Burris similarity. And generally, these take the form of measuring similarity between two kernel matrices, which in the linear kernel cases would be like distance functions on the matrices XX transpose and YY transpose. And so these RSA-type methods are very popular in the literature and are practically used to compare sets of neural representations. But these methods have been hard to interpret given that they don't explicitly comment about the neural geometry picture. And so overall, this collection of methods for comparing similarity is really confusing, and these two categories are often seen as like separate approaches to this problem with little understanding of whether they actually agree with each other and under what circumstances. So understanding these different metrics and how they relate to each other isn't a frivolous theoretical exercise, since these similarity measures are all actively used to compare models and neural data. And so the insight provided in our paper is that there's cases where these two approaches are actually identical, namely when we look at shape distances and Burris distances. And so to that end, in our paper, we focus on three different methods. The first comes from our alignment category of methods, and that is shape similarity, sometimes called the angular Procrustes similarity, which is related to Procrustes distances. And the idea here being that we should fit a high-dimensional orthogonal transformation, which you can visualize as a rotation or a reflection, to maximize the inner product between X and Y, with the denominator normalizing this to be between 0 and 1. And independently, we have methods from the second category that compare stimulus by stimulus kernel matrices, like the normalized Burris similarity, which draws from quantum information theory and metrics on positive semi-definite matrices, and also the center kernel alignment, which is essentially the normalized Hilbert-Schmidt inner product between two kernel matrices. And so naively, you might expect that maybe NBS and CKA behave similarly, since they are both similarity measures on kernel matrices, or even that CKA might behave similarly to shape similarity, because if you squint your eyes, they kind of have similar form. However, in a way that's not really obvious at all from what I'm showing you, we find that you can actually show that shape similarity is mathematically equivalent to the normalized Burris similarity. And this leads to some interesting implications, including allowing us to generalize shape distances to cases of unequal neurons in each data set, easing the analysis of asymptotic cases, and connecting Procrustes and shape distances to extensive and well-developed literatures of optimal transport theory and quantum information theory. Also, of more practical importance, one will find that for large data sets, there can be serious advantages to either the shape similarity picture or the RSA-style picture of this same similarity measure. For example, for a single-cell RNA sequencing data set, which consists of large matrices of size maybe 6,500 genes counted in 2,500 cells, and one of these for each animal, I might be interested in quantifying the similarity between the high-dimensional gene expression profiles over cells between two animals using one of these similarity measures. And I can compute this for shape similarity and also for normalized Burrs similarity. I'll see that they give the exact same number, obviously, to many decimal places, but with vastly order of magnitude different runtimes. And so this runtime discrepancy only grows with the number of genes or stimuli, and this could be, of course, formalized in terms of the different computational complexities of the two computations and the two different pictures. And then lastly, we analyzed the relationship between the more commonly used in the machine learning literature, machine learning and deep learning literature, center kernel alignment, and the now unified NBS and shape similarity. And so what we find is empirically that the NBS and CKA scores can differ substantially, especially as a function of the rank of the kernel matrices. So we find that you can bound NBS in terms of CKA with their relationship becoming less determined as the rank of the kernel matrices increases. So in the more practical case, they're less and less, they constrain each other less and less. And these bounds are shown in this left-hand figure for some low rank random covariance matrices. So you can see the bounds. And we can also see this looseness between NBS and CKA scores in real data, as here are some similarity scores between the 29 different animals for the single-cell RNA sequencing data set I mentioned earlier, and we can see that NBS and CKA don't have a one-to-one relationship, which could lead to different scientific conclusions depending on which similarity score you're using. And so for some very interesting subsequent work that I wasn't involved in at all, but I think that makes this point better than I do, you can see this paper by Kloos et al., which I think is going to be presented here. And they show with really careful experiments that these similarity measures and also linear regression generally don't agree about the similarity of models to neural data sets. So definitely interesting to check that out. And so just to summarize, what we are presenting here was we found a theoretical bridge between two seemingly very different methods of measuring representational similarity and an empirical discrepancy between some superficially similar methods. So I think that just most of all motivates that we need to think carefully about this landscape of similarity metrics that we've created, and because there can be unexpected connections and then also disagreements. So for more, I'd like to acknowledge my mentor Alex Williams and collaborator Brett Larson, and for more information on our mathematical details, including some more mathematical results on this topic, so you can see our paper or our poster, which is on Thursday. So thank you for your attention.
Hi, thank you for your talk. So what I wanted to highlight is that at the end of the day, we want to find the right way to infer neural properties from these measures and to compare things across scales and techniques, things like that, what RSA sort of aims to do. Mm-hmm. And the fundamental issue here is that the space is anisotropic. And the direction that has to do with signal strength or energy needs to be distinguished with structured signals. So none of these methods acknowledges this fact, that the space is translation and rotation sensitive. Therefore, the characterization of the structure of the space with respect to the meaningful origin is fundamental. So what do you think about incorporating this physical constraints into, or thinking about inferences about representations?
Okay, so I think what you're getting at is whether or not similarity measures are actually measuring something computationally interesting about the representation, which is definitely an interesting, no? Okay.
No, what I'm saying is that if you have a signal that has an energy state, the direction of a vector in that space tells you something very different than its length. Therefore, characterizing representations as geometric objects that you rotate and translate is misleading. There is knowledge from physics that constrains the nature of the problem.
Oh, yeah.
The space has a radial structure where the origin has a meaning. So none of these things is considering this fundamental fact, which I've been highlighting for 10 years. So I was wondering why.
Okay, yeah, so you're talking about the rotational invariance as like an ansatz that's a nuisance transformation we're taking out is built into a lot of these, like the Procrustes systems, for example. The Procrustes kind of, generalized Procrustes metric setup is nice in that respect because we can posit a different, like a group of nuisance transformations and invent other metrics that are invariant to things like permutations in neurons, so that respects the neural basis and the firing rate tuning curves?
No, it does not. That's the point.
Okay. I don't think I understand your question. So we can talk.
It's important to understand, so tuning functions, okay, forms of tuning or spatial structure in the case of a larger scale pattern changes with rotations and translations, especially due to the length of the vectors, their norms. And the energy of those signals is very different from their structure, and none of these methods captures that. So what I'm saying is we need to incorporate knowledge about the structure of the space in order to start inferring things about those networks. And these methods just translate and rotate like Procrustes, or any methods that ignore the anisotropy in the space are just misleading. That's the point I'm trying to make.
So inventing different metrics that don't involve like or the orthogonal symmetry group, or the orthogonal group is definitely interesting, and we have some work on that involving like permutation invariance as well. But yeah, we can talk. We're also doing a tutorial later in this afternoon on kind of similar topics, so maybe check that out.
Talk later. That's okay. Thank you.
All right, let's thank Sarah again. And let's thank all 5 of our speakers in the session. We have a short break right now, and we'll resume again at 11:45 with our next keynote talk.